Yeah, good afternoon, folks. This is Juergen from CSAT. Today I want to discuss a little bit about Spanish flu, the influenza pandemic, which happened in 1918. It was an H1N1 influenza virus, which was responsible for the Spanish flu, and it killed between 20 and 50 million people. The number is not correct, not clear, but people believe at least 20 million people, so more than in World War I. And the epicenter, people believe it started actually here in Kansas, not far from here, in a place right now called Fort Riley. It's a fort for the Army. A lot of soldiers are stationed here. And I believe it was called Fort Funston at the time when the flu broke out at that fort in 1980. Why do I discuss Spanish flu today? Because we have this conference coming up in May, May 7th through 10th, and we offer a tour for participants to go to Fort Riley and see the historical site, to see an historian will talk to us about Spanish flu and how it affected Fort Riley. And I believe I want to talk today, it's a very fascinating story, because the Spanish flu virus, the 1918 H1N1 virus, was the first infectious agent rescued from death. In other words, nobody could isolate that virus. I know people who went to Spitsbergen, to Alaska, and dug out corpses from 1918 in the permafrost, took samples from the lungs, took them back to the labs and tried to isolate the virus. Was not possible. So what the then researcher at the AFIP, American Forces Institute of Pathology, AFIP, did, Jeff Taubenberger, he found paraffin blocks with lungs from people who died in 1918 with flu. And then he was able to isolate the nucleic acid out of these paraffin blocks and then use this nucleic acid to amplify each individual influenza gene segment and could puzzle it together. So the entire sequence actually of from two people which died of Spanish flu was sequenced that way by using paraffin blocks from lungs which were first fixed in formalin and then paraffin embedded, and isolated nucleic acid out of that, and then puzzling the genetic code of influenza viruses together. Then my collaborator and friend Adolfo Garcia Sastre from Mount Sinai School of Medicine in New York City, he took the sequences and used a novel method called reverse genetics and put them together and transfected this with special technologies into cells, and lo and behold, after several days, a virus grew out of these cells, and the 1918 Spanish flu H1N1 virus was recreated in the test tube. Studies were done then with this virus in mice, monkeys, ferrets, pigs, dogs and cats, and to make a long story short, mice, monkeys and ferrets died after infection with 1918 Spanish flu, a high mortality rate. I was involved with studies in pigs, ducks and chickens together with a team in Winnipeg, the Canadian Food Inspection Agency, and we did the studies in the BSL-4 environment, and neither pigs nor ducks nor chickens died. So anyhow, 1918 Spanish flu virus killed 20 to 50 million people, the first agent ever rescued from death by using modern technology done by Adolfo Garcia Sastre and then tested in various animal models, was still deadly for many animals, but not for pigs and also not for poultry. Thank you very much.